Our next speaker is Adrian Liston from University of Leuven.
Okay, thank you very much. What I'd like to talk to you today about is activation of the inflammasome. So the inflammasome is really this key structure in the cell which assembles during inflammation and is absolutely essential for activating the innate inflammatory process. One of the key functions of the inflammasome is the cleavage of pro-caspase 1 into an activated form of caspase 1, which then in turn can cleave pro-IL-1 into an activated form of IL-1. So when the inflammasome has been assembled, you're able to produce mature IL-1 beta and really drive a local inflammatory response. Now, one of the molecules that can start the activation of the inflammasome is pyrin. So pyrin is one of these pathogen-sensing molecules. It's clear that it can sense pathogen products. The best known of these is the toxin TCDB, and the addition of the toxin TCDB results in pyrin activation. So this is a great paper from Feng Xiao's group, which is looking at the components of the inflammasome in a normal control cell. If you add TCDB, you have this diffuse monomers assemble up into these tiny macromolecular structures, which is the active inflammasome. And then the active inflammasome can drive the inflammatory process. Now, the really interesting thing about this is that most pattern recognition molecules or infection sensors are sensing, directly binding and sensing the products of the infection. And yet, in this paper from the Xiao group, it showed that it wasn't really the structure of TCDB that was required to trigger pyrin activation, but rather the enzymatic activity of the toxin. So this implies that pyrin is a pathogen sensor, but not in the same terms of, say, a TLR or an NLR, which are directly binding bacterial products. But it can somehow sense that there's a pathogen. It just doesn't seem to be acting in a direct mechanism. But really, the idea of what pyrin is actually sensing has been quite elusive. Despite this, pyrin is actually one of the most important molecules in the inflammasome activation. And we know this because the mutations in pyrin cause familial Mediterranean fever, which is the most common autoinflammatory disease. So this is a structure of pyrin, and you have multiple different mutations throughout the structure that are associated with Mediterranean fever. Now, Mediterranean fever is a disease where the patients will get quite short episodes, just 1 or 2 days, of fever, abdominal pain, joint pain, and this can progress into keratosis if untreated. But the molecular mechanism by which these mutations in pyrin are causing autoinflammatory disease have really been unknown. We don't know much about the molecular regulatory system of pyrin. Now, I started studying this in the context of a large Flemish family that had a familial neutrophilic dermatosis disease. So this is the Flemish family tree. Later on, we also studied several smaller families in France, Lebanon, and the UK. And the blue individuals here are the affected individuals which have an autoinflammatory disease. This is a very strong autoinflammatory disease, but it's quite different from familial Mediterranean fever. The episodes of fever and inflammation are very prolonged. They're weeks and months rather than just a couple of days. They also don't develop into keratosis. And the most striking thing is that they have this really severe skin disorder that's completely missing in the case of familial Mediterranean fever. So this is the skin disorder. In the least severe cases, you have what looks basically like a severe acne, while in the most severe cases, you get these skin lesions that really just take months or even years to heal up under intensive treatment. So these skin lesions just keep on growing and being maintained as this inflammatory lesion. If we look at the biopsies of these inflammation, they're full of neutrophils. A lot of neutrophils are in the inflamed skin. There's also joint pain, and one of the patients developed such inflammation of the heart that they needed a heart transplant. In that case, the muscle actually seems to be infiltrated with monocytes rather than with neutrophils. But in both cases, we've got this tissue infiltration of innate cells. So since we were lucky enough to be studying a family with multiple family members, quite a clear dominant inheritance of the mutation, we're able to do a classical linkage analysis. And if we do this, we can see that there is linkage on chromosome 16 in the area around Pyrin. The G for Pyrin is MEFV. And then we're able to do all exome sequencing and home in on this section. And in this region of the chromosome, the only mutation that caused the coding change was in MEFV, in the gene that encodes Pyrin. And this is a mutation in the serine residue at 242. This work was done by Vasiliki Legou. And at that point, we wanted to work out what it is that's happening, why this serine residue, 242, is causing a dominant form of inflammatory disease, unlike all the other mutations that cause a recessive form, and they cause a different disease. They cause familial Mediterranean fever, rather than this neutrophilic dermatosis. So for this, we collaborated with Seth Masters from WeHi, and we wanted to look at the biochemistry, the biochemical effects of this Pyrin mutation. So the two main things that the inflammasome activation will do is cause the production of mature IL-1 beta, or the activation of the pyroptosis pathway. So if we look at the situation here, what we've got is we are transfecting in cell lines with either a control plasmid, a Pyrin plasmid, this is wild-type Pyrin, or the S242R plasmid. And if we have a look at this, we see that normally we don't get much IL-1 beta production, even if we transfect in the Pyrin. If we add in the S242R Pyrin, then we're getting now spontaneous production of IL-1 beta. This is not dependent on endogenous Pyrin, because if we knock out endogenous Pyrin, we still see plenty of IL-1 beta production, but it is directly dependent on caspase 1. So it is working through the classical inflammasome pathway, which is caspase 1 dependent. And the same results more or less hold up true for pyroptosis. So if we analyze S242R by mass spectrometry, what we see is that we are missing a phosphorylation site at S242. So normally this residue comes up as being phosphorylated, and of course the phosphorylation site is not present anymore because you don't have the serine there to be phosphorylated. So we're removing a phosphorylation site. The effect of this is that we're missing the binding of one binding partner, 1433. So this is the pull-down experiment and then running a Western blot, and this is where Pyrin is running, and we're missing just this really small band down here. But if we cut out that band and do mass spec, we see this band is constituted of a number of different family members of the 1433 family. So the 1433 family is a family that is able to bind phosphorylated serine residues. So this seems to be bound to the wild-type form, but it seems to be missing from the S242 form. And this is now with a GST pull-down experiment. So here we can see if we pull down GST Pyrin, and then we probe for 1433, we have strong binding. If, however, we have S242R version, we don't have any binding of 1433. So even though there are a couple of different phosphorylation sites on Pyrin, this one particular site is completely required for the binding of 1433. So to try to explain how we're getting disease in this situation, we came up with a model where 1433 is acting to shut down Pyrin activation. So in a normal situation, we don't get IL-1 beta production for two reasons. First of all, we don't have pro-IL-1 beta in the system because IL-1 beta is not transcribed in its baseline state. But secondly, Pyrin is going to be locked down. And our hypothesis is that this lockdown occurs because of the phosphorylated residue, which then binds 1433, and 1433 basically sits on there as a safety catch on Pyrin. This means that even if we get pro-IL-1 beta in the system, such as by adding LPS, this pro-IL-1 beta is not going to be turned into the mature form because that Pyrin is locked down so the inflammasome does not form. Now, if this hypothesis is correct, then bacterial toxins that can activate Pyrin, such as TCDB, they could be working by removing the phosphorylation site, which would allow 1433 to dissociate. And in that case, you've now taken the trigger off of, you've taken the safety catch off of Pyrin, so now you're able to have the activation of the inflammasome and conversion of pro-IL-1 beta into IL-1 beta. So to look at this, we did the same experiments, the GST pull-down, except now on the right, what we've done is we have taken either the wild-type Pyrin, untreated, which has 1433 bound to it. Now, if we add TCDB to this, to these cells, again, we're dealing with the wild-type Pyrin now, if you add TCDB, you're completely missing this 1433 binding. We're also missing the phosphorylation site, but most importantly, we're missing that 1433 binding, which means we've taken the safety catch off of Pyrin. Now, the effect of this is that we're going to be now activating the IL-1 beta production pathway. So in this graph, we're looking at monocytes from either healthy individuals or from patients. And if we just look at the patients in black, the IL-1 beta production, of course, there's not much IL-1 beta production in the control status. Here we were very careful to use LPS-free media. If we just add LPS, we're getting pro-IL-1 beta, but we don't get mature IL-1 beta because we don't have activation of the inflammasome. If we activate Pyrin alone, we don't get any IL-1 beta because we don't have the pro form made. But if we activate both, that's when we get nice production of IL-1 beta. Now, if we look at the patients, on the other hand, we see a different phenotype. The patients don't have any spontaneous IL-1 beta production, but all we need to do is add LPS into the system. And now the LPS, which makes the pro IL-1 beta, that is going to be automatically processed into IL-1 beta. So you don't need to have the activation of TCDB. And TCDB itself doesn't do anything further on these cells. So if you're missing that serine residue, then adding TCDB is not enough to have an effect on spontaneous IL-1 beta production. So this is our model then of what's happening in these two diseases caused by Pyrin, familial Mediterranean fever and neutrophilic dermatosis. In the context of familial Mediterranean fever, it's clear that there are mutations in Pyrin that make Pyrin more active. However, we still maintain the phosphorylated site on the serine 242, and which means that the Pyrin is still locked down by 1433. So in the baseline state, we don't have Pyrin activation. However, if we do get some type of activation, for example, a bit of LPS and TCDB into the system, we are going to get the dissociation of 1433, and this Pyrin, once it becomes activated, is going to have slightly prolonged activity. Really, the molecular effects of the familial Mediterranean fever mutations haven't been well defined, but it does seem like all of the regulatory networks are intact, and it's just that when it gets activated, you have a bit extra inflammation, which is why most of the time these patients are healthy, but when they get inflammation, they have a couple of days of more severe disease. In neutrophilic dermatosis, however, we think there's quite a different situation. Here, we have a situation where the residue that should be phosphorylated is mutated, which means 1433 is just constitutively absent. We're missing this entire regulatory system, so we're always going to be getting this activation of Pyrin and the inflammasome. We're still not going to get spontaneous IL-1 beta production everywhere, though, because we still need to have something stimulating pro-IL-1 beta production. But as soon as that happens, for example, on the skin, you're going to be exposed to commensal LPS. You're going to get pro-IL-1 beta, and then that's going to be causing really a chronic activation of the mature form of IL-1 beta, which is why I think that these patients get really these chronic skin inflammatory conditions, unlike the familial Mediterranean fever mutations. So this is just the two conclusions I'd like to make. First of all, Pyrin is a pathogen sensor, but it is not like the known mammalian pathogen sensors. It doesn't seem to be directly sensing a bacterial or pathogen product. Instead, it seems to be sensing the cellular phosphorylation balance. This is similar to the guard hypothesis in plants, and I think the most important part of this is that it indicates that we may have a hypothetical cause for sterile inflammation that happens in sterile inflammatory diseases, such as neurodegeneration. In that case, anything that's going to be disturbing the phosphorylation or dephosphorylation of Pyrin is going to be triggering the same activation of the inflammasome as a bacterial toxin. So it will work via a sterile trigger. And I think this has also now been shown. There's an elegant paper just recently on NOD2 being triggered by ER stress. So I think more and more we're going to see that some of these inflammasome pathogen sensing molecules are actually sensing disturbances in cellular homeostasis. And by this, they're able to respond the same way without necessarily directly seeing the bacterial or pathogen product. Now, the second point is much more relevant for the patients, and this is that this mutation, if we're correct, should be driving the neutrophilic dermatosis via the production of IL-1 beta. So for this, we collaborated with Sinsa Savic from Leeds University, who had one of the patients in the British cohort, and this patient really had chronic elevation of inflammatory mediators. It was completely resistant to methotrexate. If we added anakinra to block IL-1 signaling, however, we saw within a couple of days a complete remission. The inflammation disappears, the skin condition disappears, and the patient remains very healthy. So thank you very much. I'd like to thank everyone in my lab, and especially Pyrin Walters, who was the key clinical collaborator on this family, and Seth Masters, who was really the key biochemist in this collaboration. Thank you.
And does the half-life of Pyrin, is half-life of Pyrin affected by the mutation? Is protein sort of basically now more long-lived? Is 1433 recruitment part of turnover process of the protein?
So we don't know about turnover in particular. The total protein levels seem more or less similar, maybe even slightly lower. So it doesn't seem to be just extending the generic half-life, but we haven't measured that directly.
Localization?
Well, localization is a little bit complicated because you do have this inflammasome process going on. So localization does seem disturbed.
Hi. Do you know how the TCDB phosphatase activity helps the toxin activity?
No, we don't. So,
Because otherwise it seems like the TCDB that's recognized Pyrin, not the other way around.
yeah, so we don't know the kinase and the phosphatase that are directly altering Pyrin. There could be multiple kinases and phosphatases that are involved. And we don't know whether or not where the toxin is inactivating a kinase or activating a phosphatase, or even creating a situation where 1433 is soaked up out of the system on other binding partners. For TCDB, there is probably going to be one answer, and I think Dan Kastner's group's been working on this and should come out with something quite soon. But Pyrin is going to be activating based on a number of different bacterial products, and it's possible that multiple different bacterial products will be activating Pyrin through altering different kinases or phosphatases. We just don't know that at the moment.
Okay, with that, let's thank all the speakers, and we'll take 15-minute break.